Hello, everyone. Welcome to the 6th online workshop. Today we'll be spotlighting the Jeff Minor Lab. It's good to see so many people online, despite the amazing weather that we've been having recently. This spotlight will be focusing on the Minor Lab. We had a very successful talk by a few members of Rachel Lennon's lab a month ago, so we're trying to build on that success. We're very privileged today to be joined by Alex Norris MP, the Shadow Minister for Health and Public Safety in the UK. Today, presenting, we'll have 5 amazing presenters. They'll be talking about their research and work to you. Let's begin with some introductions. For over 25 years, Jeff Minor and his lab members and collaborators at Washington University have been using mouse models to investigate genetic diseases of the kidney, including Alport Syndrome. The lab is using state-of-the-art mouse gene editing and high-resolution imaging to better understand what happens in the kidneys affected by Alport Syndrome. It's collaborating with drug companies to test promising new therapies that could someday be used to slow kidney disease progression in Alport patients. The first of our speakers is Jeff Minor, the head of the lab and the professor of medicine, presenting his work using mouse models to understand Alport syndrome progression. After that, we'll have Stephen Funk, an instructor in medicine, who will be talking about the effects of filtered albumin and fats on Alport mice. Our third presenter is Hani Suleiman, an assistant professor of medicine, and he'll be presenting his work imaging podocyte changes in Alport syndrome. Penultimate presenter is Liang Ning, a staff scientist who will explain how the podocyte cytoskeleton could be a potential target for efforts to delay the progression of the disease. And our final presenter today is Kohei Umachi, a postdoctoral research scholar whose presentation is titled A Personalized Medicine Platform for Overcoming Nonsense Mutations in Alport Syndrome. As I said, I'm joined by a scientific moderator who will help put some of the questions to these speakers. This week, that's Neil Turner, the professor of nephrology at the University of Edinburgh. Right, I think that's about enough from me. It's time I hand it over to Jeff to kick us off. Jeff, would you mind sharing a screen? Oh, he's already there. Perfect.
I beat you to it.
You did. Go for it.
Well, thanks so much, Patrick, for the introduction. Thanks to Susie Gear and Alport UK for inviting me and my greater group to present some of our recent research on Alport syndrome, which, as Patrick indicated, I've been studying for over 25 years. And I'm unable to advance my slides.
Change. We didn't have this at the start.
Not sure what's going on. I guess I'll use my mouse. Okay.
Oh, there we go.
So first, I want to point out that when I give international talks, I have to tell people where Washington University is. It has a strange name because we have Washington, D.C. over here, where George Washington University is located. We have the state of Washington, Seattle, where the University of Washington is located. But Washington University in St. Louis is here in the middle of the country along the Mississippi River, just to give you some perspective. So what I'm going to do is give a little bit of an introduction to kidney function, what goes wrong in Alport syndrome, and touch on brief points that the next 4 speakers will be discussing. This might be review for some of you, but I think it's important to make sure we're all on the same page of understanding, at least for today's talks. So the kidney is composed of nephrons. Each nephron is a single filtration unit that has a glomerulus followed by a tubular portion of the nephron. The human kidney has about a million of these individual filtration units, nephrons. And in chronic kidney diseases, over time, these nephrons begin to lose function. And as these nephrons lose function, the glomerular filtration rate decreases. And if that decreases too much, then there begins the decline of kidney function, eventually leading to end-stage renal disease. So here's a more detailed and higher power look at the schematic structure of the nephron. This is the glomerulus, which is affected by the mutations that cause Alport syndrome. This is where the kidney filtration barrier is that actually does the filtration of the blood. That primary filtrate leaves the glomerulus and flows down this tubular portion of the nephron, where the cells that line that nephron change the composition of that filtrate by removing compounds that the body wants to retain, but leaving those waste products that we want to get rid of as the urine. So as that filtrate flows down here, it becomes concentrated, leads to a collecting system that eventually takes it to the ureter and then down to the bladder, where we hold it until we can eliminate it in a proper fashion. So I wanted to show you this video that was made by some researchers at Indiana University in Indianapolis, where they did live imaging of glomerular filtration in a live rat kidney. And they basically exposed the kidney through the back and stuck a microscope objective on top of it, and they injected fluorescent substances into the bloodstream. And let me just start it as I speak. And they injected large substances that are yellow, or that fluoresce yellow, and then small substances that fluoresce red. A mixture of the two into the vein of the rat. So those substances make it rapidly to the kidney. You can see these are the glomerular capillaries, which are glowing yellow because they're filled with this substance. But you can see around the capillaries, it quickly becomes red because the red small molecules are crossing the glomerular filtration barrier and then flowing down the tubular portion of the nephron, which is represented in the schematic here. But these larger substances, which are yellow, are retained within the capillaries. So you're seeing them in the glomerular capillaries, you're seeing them in the other capillaries that surround the tubule, as shown by the blue and red tubes here. So the whole purpose of the glomerulus is to be selective, to allow the small molecules to cross the barrier, but to retain the large ones. And most of the large molecules are actually proteins. So when there's proteinuria, when protein appears in the urine, especially albumin, that means these large molecules are for some reason able to cross the filtration barrier. And there are many glomerular diseases, including Alport syndrome, that have proteinuria as a major problem. So this demonstrates what's called glomerular perm selectivity. So that filter knows how to normally not allow large molecules to pass, but to allow small molecules to pass easily. So here's a schematic diagram of the major components of the glomerulus. You're now seeing a cross-section of those capillaries. The basement membrane, the glomerular basement membrane that's impacted by Alport syndrome mutations, is this blue, smooth, ribbon-like structure, which has a cell, a very specialized cell that will be talked about today called the podocyte, sitting on one side of it, and it's lined by another cell type called an endothelial cell. And it's these unique podocytes which actually make the collagen that's affected by Alport syndrome and really should be the target of many therapies to delay or even perhaps someday cure Alport syndrome, because this is the cell that makes those collagens. And it's called a podocyte. Podo is, I think, Latin for foot, or maybe it's Greek, I'm not sure. But these cells send out processes that in cross-section, well, first let's start with this view here. You can see the podocyte has a rather large cell body where the nucleus of the cell would be, but then it sends out these major processes that subsequently branch and form interdigitations with processes from a neighboring podocyte that you can't see the cell body of in this particular view. But if you look at this in cross-section, you can see these foot processes are sitting on the glomerular basement membrane, which has the collagen that are defective in Alport syndrome. And this is basically what formed the glomerular filtration barrier. There's the endothelial cell here adjacent to the blood. There's the basement membrane where the collagen is located. And then there are these podocyte foot processes, which are linked to each other by a very specialized structure called a slit diaphragm, which is important for maintaining the efficient flow of filtrate from the bloodstream into the urine, the urinary space. So those red molecules would easily cross here in the movie I showed, but the larger yellow molecules would remain within the bloodstream because of the size selective properties of the glomerular filtration barrier. So that glomerular basement membrane doesn't just have this important collagen 4 trimer, which consists of the alpha 3, 4, 5 chains, each encoded by a gene that when mutated causes Alport syndrome. There are other proteins called laminin, nitogen, and agrin. Hani in his talk will mention agrin. that we use as a landmark in some imaging studies. And then Kohei in his talk will mention how mutations that prevent production of the full-length versions of these proteins, which in some cases are nonsense mutations, can cause Alport syndrome, and how we might be able to get around that type of mutation. Because if any one of these three chains can't be synthesized properly, or might have a mutation that causes a defect in part of the chain, that will result in Alport syndrome. So in Alport syndrome, the basement membrane, rather than having this nice, smooth, ribbon-like appearance, takes on this split, thickened, and kind of lucent-looking appearance, which indicates that there's a significant problem. And this type of appearance of the basement membrane is almost never seen in any other disease except for Alport syndrome. So it's a really good indicator that this patient would have Alport syndrome. Of course, this patient is actually one of the Alport mice that I made back in the 1990s and published in 1996. And this is sometimes referred to as a basket weave type of appearance, because it looks like there's somewhat of a meshwork of these basement membrane proteins forming within the space of the basement membrane. Now, these changes don't all happen at once. Early in the disease process, the basement membrane has some normal areas, and you can see some kind of slight bubbling of the basement membrane or outpocketing. And you can also see that the podocytes have, for the most part, some, but not all, normal foot processes. But later in the disease state, you can see the basement membrane becomes very thickened, and the podocytes have lost much of their foot process architecture. They appear to actually be invading the basement membrane. And when one thinks about treating Alport syndrome, I think many studies that have been done, whether drugs that others have done or genetic studies that we have done, it seems to be important to treat to prevent this from happening as early as possible. There does come a point, perhaps at this stage, where for this particular glomerular capillary loop, it might be beyond the point of rescue. Whereas in this case, there might be a way to stop the progression from going from here to here. So I wanted to give a bit of an analogy. I was going to use props, but I think it's better to show you actual pictures. So the normal basement membrane, and this is a balloon actually, is very smooth. And if you think of these as the capillaries that are emanating from the center of the glomerulus, kind of cross sections where the blood is flowing through here, the podocytes are sitting on the surface of the capillaries, right on top of the basement membrane, represented by the balloon. And you can see it has a nice smooth surface. I think you all know what balloons are. But think of the Alport basement membrane as more of a bubbly, bubble wrap type of appearance. So the podocytes that are sitting on top of these bubbles, we think that the material present in the bubbles is not the normal basement membrane composition. And there are studies that others have done showing that the Alport basement membrane might be softer than it should be. So many of, if not most of the cells in our body are used to having a certain substrate that they sit upon normally, and they're used to that. They know how to behave on that. And stiffness is one of those qualities of the substrate that determines how cells behave. So podocytes sitting on this abnormal surface, which is likely softer, seem to not be able to behave normally. And that's one aspect of the disease that you'll hear about today. And the reason podocytes might and other cells would behave abnormally on the wrong substrate or the wrong stiffness of the substrate is because, like humans, we have our own skeleton that allows us to walk around and do things we need to do. Cells also have their own cytoskeleton, their own skeleton. It's called a cytoskeleton. Cyto means cell. And the cytoskeleton is not as stiff as bones, but there are different cytoskeletal proteins labeled here fluorescently that allow the cell to take on a certain shape. And the cytoskeleton is extremely important for how podocytes are able to elaborate those processes and maintain those foot processes that are so important for maintaining glomerular filtration And this is an aspect of podocyte biology that Hani and Liang will touch upon in their talks. So this is another schematic that I helped draw for a review that appeared in a large textbook, just to show you how the podocyte foot processes are sitting on the glomerular basement membrane, which contains the collagen alpha 3, 4, 5 chains, as well as these other basement membrane proteins. They have special receptors on their surface that allow them to link tightly to it. And they have the special slit diaphragm that forms between foot processes that is also important for this linkage. And what we know from multiple studies in mice as well as in human diseases, that mutations that affect any or many of these proteins, if not most of them, can cause glomerular kidney disease. And that's because there seems to be this rather large complex of proteins linked to proteins, linked to proteins, linked to the basement membrane that allows this filtration barrier to remain functional for 80 to 100 years, depending on how long people would live. But those that have mutations in some of these proteins, of course, will be unable to maintain this structure, and that will lead to glomerular disease and eventually kidney failure in many cases. So finally, I wanted to point out that although the genetic defect in Alport syndrome begins in the glomerulus, other parts of the kidneys become injured that helps lead to that reduction in glomerular filtration rate, or eGFR, and increases the level of creatinine in the blood. So that's because, or one hypothesis we're testing that Stephen Funk will talk about, is that substances that should remain in the blood but leak out in Alport syndrome and perhaps other glomerular diseases, they flow down with the filtrate down this tubule, and those tubular cells are not used to seeing those large proteins. And so that might somehow lead to injury in other parts of the nephron besides the glomerulus, where the Alport mutations have the primary effect. And so that might lead to injury of these other parts of the kidney and eventual chronic kidney disease. So that's my introduction for today. And this is a shot of my lab out on a lunch outing, I believe last summer. And I would be happy to take any questions, and thank you in advance, or thank you for your attention.
Thank you very much, Jeff. And good to see some of the work that your lab is doing. We're going to skip over questions for you. I want to invite you to jump in if you have anything to add over the subsequent Q&As. But for now, we're going to move on to the second speaker, Stephen Funk. Would you mind sharing your screen? Okay.
Unmute.
I think we can hear you. Okay, yeah, go for it.
Okay, so I'll just jump right into it. Thanks, everyone. I'm going to be talking about a very new project for me and for the lab. And this is sort of a first run to a crowd with this talk in a short format. So if anything comes out strange, feel free, of course, to ask for clarification. So I'm picking up some work from a previous project that was performed by a nephrologist in our division and a friend of mine now named George Gerard, who is up here on your left. And George is mostly in the clinic now and not as much in the lab. But published a few years ago now, the work George did discovered that when albumin was deleted from Alport mice, that the Alport mice had a reduced loss of kidney function. In other words, their kidney function remained healthy for a longer period of time, indicated by the blood levels of blood urea and nitrogen, which is sort of similar to creatinine. When GFR is reduced and creatinine increases in the blood, blood urea and nitrogen is just another way to measure that. So the function of the kidney was maintained for a longer period of time. And additionally, the mice lived for a longer period of time, as you can see by the black line in the right panel, indicating the Alport mice that don't have albumin. And George discovered that this elongation of lifespan was true for two different types of mice, different strains, one called C57 Black 6 and another called 129. So the big question, I'm sorry, getting ahead of myself. So the punchline to that story was sort of an answer to a longstanding question. Does albumin, when it leaks through the capillary wall, as Jeff just described, among other proteins, and in the case of Alport, red blood cells, does albumin injure these cells? And this was never done before, the deletion of albumin. And so it provided sort of a final answer to that big question. And in the Alport context, yes, it definitely does. So how could albumin injure cells? So as Jeff mentioned, a lot of things leak across the glomerular capillary wall in the context of kidney diseases and glomerular diseases like Alport syndrome. I have a cartoon here that lists a few of the ones that are very well appreciated. Albumin has actually been studied for a very long time for causing this, given that it's one of the first proteins to leak across that structure. It's also the most abundant protein in blood, and so it stands to reason that it could potentially injure the cells that don't see it very much. Other things are also known to injure the cells in the glomerulus and in the tubules downstream, such as antibodies, components of red blood cells, molecules called complement that assist in the clotting of blood, and other molecules that bind iron. So the outcome to these injuries typically results in inflammation to these cells, which can cause cell injury, causing them to not function properly. In an extreme context, it can cause these cells to die and they have to be replaced by their neighboring cells. And additionally, through this process and over time, the cell death and the cell injury can also cause fibrosis, which many of you probably know is basically sclerosis of the tissue, which leads to decline in tissue function. So which would, in the case of the kidney, would cause decreased GFR, decreased ability to manage blood and blood balance and blood pressure and things like this. So how could albumin do this? There are many hypotheses. A big one that people study is called albumin overload, where too much protein, too much albumin gets in these cells and just causes them to not function properly. Another hypothesis that is not very well explored is that albumin is a molecule that carries lots of other things. In particular, and I have these lists, and I don't mean for you to study or read through all of them, but just to show how many things albumin can carry, including common drugs that we all know, like ibuprofen, acetaminophen, things like this, actually bind to albumin and get carried through the blood. But one of the major things albumin carries is fatty acids. So as part of our diet, we take in lipids, and these are processed by the liver. And in the liver, some of these lipids, the fatty acids, bind to albumin, and albumin carries them through the blood, and this is normal physiology. And part of that role is albumin delivers fatty acids to cells where they serve many purposes, and the cells grab the fatty acids off albumin. They use it for energy, they use it for communication inside the cell, proteins to proteins, and are part of the normal function of how cells work. But in this case, a lot of albumin is spilling over out of the glomerulus, an abnormal quantity that the podocyte cells are not used to, and that the epithelial cells in the nephron tubule are not used to seeing. And we hypothesize that these fatty acids that are on albumin are part of sort of the conundrum and the injury that occurs in the glomerulus, to the cells in the glomerulus and the cells in the nephron. So borrowing some paradigms from diabetic context, where the effects of lipids are very well studied in the kidney, here's sort of an overview slide of how these lipids could be affecting glomerular cells and tubular cells in the Alport context. An overabundance of these lipids can cause abnormalities in energy production. It can cause stress to different subcompartments inside the cells. It can affect the way cells proliferate. It can cause these cells to die because there's too much. The lipids can directly cause production of oxidative stress, in other words, inflammation. And it can promote.
Hey, Stephen, sorry, we're close to running out of time. Just final.
And this is my last slide. Thanks. In addition, and podocytes specifically, it can cause changes to that slit diaphragm that Jeff described earlier, which is a critical component to mediating filtration of these proteins and keeping that glomerular basement membrane structure intact and filtering things properly. So it's very preliminary, and we're just getting this project on its feet. What we hope to do in the near future, just a few preliminary experiments, is inject free fatty acids into the mice that are labeled, and we can see by microscopy, and then see what kind of cells they're going into. We want to do high-fat diets and also fasting diets to see if that has any effect on the albumin phenotype and progression of the disease. We plan to do sort of large-scale DNA expression analyses after giving albumin back to the mice in a very stable, long-term sort of fashion. And we hope to do sort of a bulk analysis of all the lipids that are in these different compartments to see when albumin is not present, which lipids do not get in to the cells, and then define, hopefully, which lipids are doing things and leading to injury, and then assess their pathways and mechanisms after that. And so that's where we are and hope to go. And I hope this is interesting for you, and that's my end.
Thank you very much, Stephen. Obviously, I've got a few questions of my own, and of course, you can submit questions using the Q&A function now. I was wondering whether there was a possible relationship, as you slightly indicated in this final slide, between diet, therefore, and this progression through albumin of the disease. Could diet help perhaps sort of slow the onset that we're seeing here?
So what I have studied of albumin and how it interacts with lipids so far indicates that there are 7 like strong lipid binding sites on albumin and something like 20 binding sites for lipids that have sort of a weak affinity. And in normal physiology, maybe one of these strong binding sites is occupied by just one free fatty acid. And there is some give and take in these binding sites of which lipid can bind. They're not specific to a type of free fatty acid. Many free fatty acids can bind to the different sites. But so it depends on what, it can depend, I think, on what is in your diet and what other things are binding to albumin and how much albumin is available to bind all these different things. So in short, I don't have a great answer for you. I'm still studying how diet and lipid content is sort of absorbed by albumin and how that's carried from, to different parts of the body. So maybe if your diet was massively skewed in one direction, or if particular lipids have very specific effects on the cells, then maybe diet could be part of the factor. But also the biochemistry of albumin and the interaction and interplay of all these different things could be extremely important.
Okay. Thank you. In the interest of time, I'm going to hand straight over to Neil. I'm assuming you have a few questions for Stephen as well. No, unmute. I can't hear you if you're speaking.
That's
there we
better.
go.
Yep. There's been some really interesting and important comments, questions in the chat, which I'm not sure there's a definite answer to them, but it'd be really interesting to hear your views on them. So, Jeff, you started by outlining how the environment for the podocyte was very abnormal, and how that may well be making the podocytes look very unhappy and dysfunctional. In the background, and not really mentioned so far, is this whole issue of what the pressure is inside the glomerulus and whether that may be contributing to its failure. And Stephen, you've come up with the third method by which the Alport abnormality might lead to kidney damage, which is that the filtered protein in some way poisons the tubules down the way after the protein leak is established. And interestingly, you can use the fact that ACE inhibitors protect the kidneys in Alport syndrome and actually in other diseases with the protein leak to support any one of those three arguments. Again, I'm not sure there is an answer, and clearly some of your research is meant to try and find out, but it'd be really interesting to hear your comments on it, and so would some of the people asking the questions there.
Yeah, so I see Jeff is not unmuting, so I'll try to do that as best I can. Of course, we know that ACE inhibition and blood pressure regulation can reduce proteinuria, and so that is obvious then that the effects that we see of albumin should be reduced. And I'd like to mention as well that it's very well appreciated that albumin and the lipids on albumin, there is evidence that both can injure the tubules, but we also see a reduction in glomerular basement membrane abnormalities and podocyte stress when albumin is missing. So it's having an effect, it would seem, in all compartments where it typically is not in high quantities. And I'm very excited. One experiment that I neglected to list in that last slide is I'm very interested to see what will happen. We may even have the data, and Jeff can chime in if so. When albumin knockout Alport mice are put on ACE inhibition. And I think the answer would be that they live, and they do actually, I'm remembering the data, they do live even longer. They live a lot longer, actually.
Yeah.
So being able to include some form of therapeutic, if we can understand how albumin and perhaps the lipids on albumin are causing this injury, then maybe we can add an extra therapeutic, potentially even a diet angle that would stave off the injury and prolong kidney function even more than the ACE inhibition, which would be a fantastic.
Absolutely, that's definitely what we want. Jeff, do you want to comment on that?
Yeah, I was just going to bring up that, so our mice that lack albumin, the Alport mice lack albumin, we put them on an ACE inhibitor. I think we had what would be the longest lived Alport mice ever. Some lived to 18 months. And the normal lifespan of a mouse is typically 2 years, they say. But I think that indicates that, so albumin is doing damage on its own, but by reducing blood pressure, that's impacting some other pathway. Because we can add the two together, and it's not just the reduction in albuminuria that is the effect of ACE inhibition. So it could be that it's reducing stress on the podocytes and reducing their cytoskeletal defects and maintaining the foot process architecture for a longer period of time.
Yeah, I think one of the amazing things about that paper, which is George Gerard's paper, which was seriously under-referenced, is the fact that these mice without albumin, the major protein in the blood of humans as well as mice, they're very healthy and they can live. So I have not seen that intervention in an animal experiment yet, but you might wonder whether reducing albumin production in humans could be a therapeutic angle. Has anyone tried that? Because it's very easy to get inhibitory oligos into the liver.
Yeah, I don't know if it might have to do with the fact that these mice never ever had albumin, that they're able to tolerate the lack of albumin because they've compensated in some way. But whenever I talk about this, I always mention that, of course, we can't remove albumin from human patients that have Alport syndrome. I think that's just an impractical thing to do. But we want to find the pathways that albumin is activating in the nephron, causing damage, and try to inactivate those pathways with drugs that might be known already.
Yeah. So it's a really interesting, it's a really interesting observation that if people, and as well as mice who are born without albumin, seem to be healthy, but we regard low albumin in the blood as a serious sign that something is wrong, and a leak into the kidney is one of them, one of the causes of that.
So I'm going to ask you to do one more question. Just, I know we've got a plethora of options, but
I'm going to ask one question just because it's a kind of background issue. And Jeff, in describing the proteins that make the glomerular filtration barrier, one question that came up, it was by someone in Manchester, actually, who you may know, who probably knows the answer. It was a question about how rapidly do these molecules turn over? Are they there for a long time, or are they constantly turning over day by day?
Yes, rapidly is the wrong term to use. They turn over very slowly. So once the collagen 4 is made, it lasts a long time. And there's a recent paper that came out showing that using some fancy protein analysis.
Yeah.
As opposed to agrin and laminin, which have a more rapid, still not rapid, but shorter half-life or shorter time in the basement membrane. So, but I think the fact that the podocytes continue to make the RNA that's necessary to make the protein throughout life. So I think there has to be some slow production of new basement membrane proteins along with degradation of the old proteins.
Okay, that may be
maintained
later on. Thanks very much, Jeff. Over to you, Patrick.
Thanks very much, gents. Our third speaker today is Hani Suleiman. Would you mind sharing your screen, please? Okay, looks like we can see it.
Do you hear me?
Yeah, absolutely. Go for it.
All right. So I'm going to talk about a technology aspect of adapting technology to the kidney diseases. If you ever took if you remember the physics, high school physics or university physics, there's something in the microscopy called diffraction barrier. So basically, if you use any microscope, you are always, when you zoom in any structure, you at some point your optics would limit you with what we call the diffraction limit. The wavelength of light won't allow you to see beyond that level. And the level turned out to be around half the wavelength of light, which the lowest one is like around 280 nanometer. And if you think about the kidney, if you think about the kidney, the podocyte and then the food processes, they are all around 100 to 300 nanometer, which is one of the millionths of a millimeter. When we started the work to adapt to the technique called the super resolution microscopy, and this technique is it was groundbreaking, that it deserved a Nobel Prize in 2014. This super resolution microscopy technique breaks the diffraction limit, and you could now see details and structures like you see in the top panel. They are using conventional microscope with the 2-color imaging, and then the lower panel is using resolving the structures using the super resolution imaging. And you could see that now you see sharp clusters in the green and in red. So we adopted this technique to image the kidney, and one of the first things that tried glomerular basement membrane. And here I'm showing you how agrin look like in a light microscopy. We were also seeing this blurry image, And we always thought that the agrin is just one layer. We see it in like a conventional microscope. But when we used the super resolution microscopy, we could notice that it is 2 leaflets located at the edges of the basement membrane. And here I'm showing you the power of this technique. So basically, if you label the glomerular filtration barrier in red, you see the outline of the podocyte, as you see it here, the EM correlation. And then in the endothelial side, inside the capillary loop. And then in blue, you see agrin. And if you focus on this area, you could now notice that you could see the whole glomerular filtration barrier that Jeff described, the endothelial layer, the GBM, 2 leaflets, and then the podocyte, the outline of the podocyte aspect. So like this technique, and we 2013 we mapped the molecules, the known molecules in the GBM, and we looked at Alport syndrome, in fact, and we could see changes in the compensation in collagen for alpha 112 instead of when the 345 is missing. So then after what our second study is like to use this super resolution technique to ask questions about the podocyte injury, podocyte cytoskeleton, and changes that happen after injury. So we looked at different disease models. So in a typical situation, and including Alport syndrome, in a late stage of Alport syndrome, you would the food process, podocyte food processes would disappear and you get replaced by one sheet of membrane. We call it food process effacement. And to understand what's food process effacement and how that affects the kidney, the podocyte function, and they compromise the glomerular filtration barrier, we use super resolution imaging, and we could see that there's a landmark on like all these injuries, including the Alport syndrome, that it's we start seeing some structure, this like candy pattern. Located like near the GBM that are not present in a healthy situation, as you see on the left here. So we are trying to understand what does the sarcoma-like structure do to the podocyte, and first how they are formed, and how, what effect do they have on what are they reflecting in the podocyte, and what effect do they have on a glomerular filtration barrier. So in this case, like we our current like hypothesis, like when we see in Alport syndrome in advanced cases, you would see the food process effacement and the sarcoma-like structure in the bottom of the food podocyte near the GBM, and we are trying to understand how can we reverse these kind of patterns to allow the podocyte to re-establish their individual food processes, as you see it here on the left, and then trying to use this technique to study the ability for podocyte from Alport syndrome to recover and re-establish their food processes. And that's my talk. If you have any questions.
Yeah, absolutely. Thank you very much for explaining that so well. I guess I'd love you to expand on this final slide that we saw on the possibility for this helping to understand treatment in terms of reversing these changes that we're seeing. What sort of application does this have in terms of expanding our understanding of potential treatments for Alport syndrome?
Yeah, so first, and so when you could imagine the podocyte when they are effaced. What's going on? Okay, where's my arrow? I'm missing my arrow.
Oh, good.
Okay,
we can't see your screen right now, but.
Oh, you cannot? Okay.
No, you can reshare it if you want to.
Let me see. I see. You see it now?
Yeah, there we go. Looks perfect.
Okay, so So when we see food process effacement, as you see it here, and I'm pointing the arrow, traditionally we thought that, okay, it's just a sheet of membrane. But when we start noticing this sarcoma-like structure, it suggested to us that there is an accumulation of actin in the bottom of the food process. And you could imagine this accumulation of actin, or like precipitation of actin, would really restrict the podocyte from re-establishing their food processes. So it becomes like a secondary hurdle that the podocyte has to overcome to go back to its normal state. And so if we want to recover the podocytes and then have them reestablish their food processes, we really want to find a way to get rid of this sarcoma-like structure of this actin precipitate in the bottom of the effaced area before they can reestablish their food processes. If that makes sense to you.
Sure. And what's next for your research, really? What are your plans for the future?
Yeah, so we are studying now the cytoskeleton. We're trying the first thing we're doing now is trying to understand what goes wrong for these first food process effacement to appear, because you could imagine this is a major change in a podocyte. So there is trigger happening, and like in Alport syndrome is a unique case that this change, this food process effacement, comes later than other nephrotic syndromes such as Pearson syndrome. Pearson syndrome is like when in a couple of weeks, in the mouse, all the food processes are effaced. While in Alport syndrome, something happened later after the GBM is changed so much that leads to the food process effacement. And on top of that, something happened after that allowed this sarcoma-like structure to appear. So we're trying to understand what goes wrong, how the cytoskeleton is responding to these cues coming from the GBM, or the changes that in the GBM leads to this changes in the cytoskeleton. So if we understand that, we will be able to intervene and stop the food process effacement. And on top of that, we want to understand, find a way to reestablish, get rid of this sarcoma-like structure and reestablish the healthy food processes.
Okay. Thank you very much, Hani. I think that's enough from me. Neil, I'm assuming you've got a few questions as well.
Yeah, sure. Hani, there's a couple of questions that are very specific, which I think you might like to look at later. But there's some really important ones about, it's really fascinating to study this model, and those are just wonderful pictures. But I think one really interesting question is, in Alports, we know what the problem is. But quite a lot of what happens in Alports and the treatments for it seem to be shared with other conditions. So I wonder how unique to Alport syndrome these changes are that you're seeing, or do you see them also in other diseases with a protein leak?
Yeah, so this sarcoma-like structure, we see it so far universally. We see it in every disease that have food process effacement. But interestingly enough, not every effaced area has it. So there is a difference between a food process effacement. Some of them, they don't have it, some of them have it. And then the difference, I think if we understand the difference between the two, we'll be able to intervene. I would imagine that the one that doesn't have this actin precipitate, they have the potential to retract and go back to their interdigitating food process shape, while the one that has this actin precipitate, they might not be able to reestablish. Does that answer your question?
Yeah, I think so. There was a question again, which perhaps relates to how good the inner test tube modeling of these diseases are. Because if you look at podocytes in a dish, they're sometimes very well differentiated and sometimes not. And if you give them various toxins, they seem to de-differentiate and lose their expression of some of their molecules. And I just wonder whether you're seeing that kind of thing here. I mean, for instance, if you give an animal ACE inhibitors, is it protecting it by directly signaling to the podocyte, or is it protecting it by slowing the rate of basement membrane damage? Alluding really to the problem I raised earlier of knowing which of several mechanisms are key here.
Yeah. So modeling podocyte in culture is not an easy thing. Like
No,
the moment you take the podocyte out of the mouse, like really change shape so much. No, now we are testing other ways of getting more in the physiological range of stiffness, like as Jeff suggested, the culture podocyte on like more physiological stiff substrate, and then the right substrate, because we always used to culture them on glass or plastic, and that's never physiological. So trying to model the podocyte in culture in a more physiological fashion, and then this out in signaling, like what goes wrong, whether it is just sheer stress coming from the like the ultrafiltrate, or it's out in signaling from the changes in the molecules. This model that we will have in culture, we will be able to manipulate these aspects and then study the podocyte in more physiological ways. So basically, culture the podocyte on like laminin or collagen for alpha 3, 4, 5, or change that to a pathological collagen. So,
but I have to say, I think your technique of looking at it in vivo, or just immediately ex vivo, is really powerful and could validate some of these tissue culture techniques, or not. Yeah, thanks very much. Patrick, do you want to move on at this point? There's one or two very specific questions, Heini, I'm going to suggest you might like to answer there about the techniques and some very specific findings about particular molecules.
I think we could answer them later. Absolutely. I think if people keep an eye on the Facebook page, we've hit our mark to move on, if that's okay for now. So thank you, Hani and Neil. Moving on to our penultimate speaker of the evening, Liang. Would you mind sharing your screen? Fantastic. Looks like we can see it fine.
You hear me?
Yep, looking great.
Okay. And I'm going to talk about our research, the podocyte cytoskeleton, its potential target to delay Alport syndrome progression. And currently, there is a lack of therapy available to show progression of kidney disease in Alport syndrome. And podocyte injury and depletion have been proven to cause progression to end-stage kidney disease. Therefore, protection of podocytes from injury is thought to be important in therapeutic approaches in Alport syndrome. And podocytes lines up the side of glomerular filtration barrier. It has a large cell body that extends the major processes, as you can see here. And the major processes branch further into food processes. In this study, we focus on the cytoskeleton in food processes. And here is the schematic of cytoskeleton molecular components inside two adjacent food processes. There are many proteins inside the cytoskeleton that functions to regulate podocyte shape, structure, and spatial organization, and provides to interact and respond to environment and the driving forces for cell motility. The cytoskeleton interact with extracellular matrix such as basement membrane, where focal contacts and the receptors. So in Alport syndrome kidney, the defective glomerular basement membrane, lack of collagen for alpha 3, 4, 5 may cause disorganized cytoskeleton and podocyte injury. And synaptobodin, the protein, as you can see here, is thought to be an important component of the podocyte's inner cytoskeleton. We found that the mice lacking synaptobodin had normal kidney function, but podocytes are more susceptible to chemical-induced injury. This prompted us to investigate how the vulnerable cytoskeleton in food processes affects the progression of Alport syndrome. Then we created synaptobodin deficient Alport syndrome mice and compare with the conventional, traditional Alport syndrome mice. We collected urine at different weight age, sacrificed the mice at 8 and 15 weeks. We performed histopathology and analyzed the urine and blood and performed electron microscopy. Here are the results. Synaptobody deficient Alport mice showed a significant reduction in survival rates compared with the normal Alport mice. Urinalysis showed a significant increase in urine albumin to creatinine ratio as early as 6 weeks, and the ratio increased as mice aged. It showed significant increase in synaptobody deficient Alport mice at 8, 9, and 13 weeks. And renal function was also analyzed by measuring blood urea nitrogen. It was higher in synaptobody deficient mice at 18 weeks of age. So here are the representative histopathology imagings. At 8 weeks, many glomerulars, and the lower panel is the synaptobody deficient mice imaging. And many glomerular sclerosis and tubular protein cast appeared in synaptobody deficient collagen Alport syndrome mice, which progressed to end-stage renal disease pathology that induces global diffuse glomerular sclerosis and crescent, as you can see here, and widespread protein cast at 15 weeks of age. In contrast, less severe pathology was observed in control litmates. The quantification of glomerular lesions showed significant increased glomerular sclerosis rate percentage. So here are the representative electron microscopy imagings. It revealed that mild glomerular basement membrane splitting, and as you can see here, and thickening and bulging. These are typical of Alport syndrome at early stage. In comparison of this, synaptobody deficient Alport mice revealed late stage pathology that include extremely thick glomerular basement membrane and a significant worse for processive fission. So all of these observations indicate the loss of synaptobody accelerate Alport syndrome progression. So we conclude that this study reveals a protective role of synaptobody in progression of Alport syndrome. It also reveals the podocyte cytoskeleton as a potential therapeutic target, and stabilization of cytoskeleton in kidney podocytes may be beneficial to Alport syndrome. That's all. Thank you for your attention.
Thank you very much. I think a lot of patients are probably thinking right now what the sort of parallel is between what you've seen in these studies and what might be happening in our bodies as well. What could this potentially tell us about a human onset of the disease?
The cytoskeleton drug only we can find in clinic is the functions to microtubules. There are 3 types of cytoskeleton: microfilaments, and microtubules, and the intermediate filaments. In this study, we focused on the actin cytoskeleton for processes. So the first thing we need to know is because the drugs lacks of specificity, so it's hard to use in clinic. But the first thing we need to know is the specificity to determine which drugs can use for cytoskeleton, actin cytoskeleton. So currently, the only thing we can do to know the mechanism in cytoskeleton and the extracellular matrix interaction.
Well, thank you. Neil, I'm seeing a question in the Q&A that I do not understand, so I would love to hear your thoughts on that.
Yeah, so Rachel Lennon has asked an important question which leads to a follow-on, which I will ask. And the question is, do mice with synaptopodin-only knockout mice, so synaptopodin-only knockouts have GBM defects, or do they just have podocyte defects?
And in our synaptopodin knockout mice, there's no defect in vivo, and but after treatment with adriamycin and podocytes, the synaptopodin knockout podocytes showed susceptible injury compared with the control mice.
Yeah. So your experiments fit really well with the observations in patients, where quite often now patients with unusually severe disease are found to have a second mutation. So Alport's patients might have a mutation in a podocyte gene. Polycystic kidney patients might have one of the other molecules involved in cyst generation with a minor mutation in it. But I think So that is a kind of important lesson. Looking at the Alport target gene alone may not be what we're doing in a few years' time. We may be looking at a whole set. But the molecules that combine to make things worse usually are related in some way, and it seems highly likely that many of them will be in podocyte-type genes. So earlier on, I posed this question about, is the key pathogenic problem here? Is it the failing podocyte? Is it the failing GBM structure, the pressure just leading to the structure falling apart? Or is it Stephen's question? Is it the toxicity of the protein downstream? Now, of course, all of these things could be true, and it's not necessarily one or the other, but probably there are some problems which occur before the other ones. In the drugs we use, or drugs that might be available, I wonder which ones stabilize the cytoskeleton of a podocyte, either in vivo, we probably don't know very much because Heine hasn't done all those experiments yet, or in a dish. Are there any drugs around that we use that affect podocyte structure in a tissue culture dish? Actually, I think
I'll chime in on that.
Yeah, please, Jeff.
I think the answer is there are some experimental, not ready for human drugs out there that are thought to impact the cytoskeleton in multiple glomerular diseases. At least they seem to affect the podocyte cytoskeleton. But those I don't know of any FDA approved drugs for humans that would target the podocyte cytoskeleton, but I don't know that people who design drugs have thought of looking at, say, a synaptopodin stabilizing drug to improve its
I don't think
interaction with the actin cytoskeleton.
I don't think there's anything that would be a synaptopodin specific drug, but certainly in tissue culture, there's lots of demonstrations that angiotensin leads to cytoskeletal rearrangement, and so does endothelin. And angiotensin receptor blockers and ACE inhibitors reduce angiotensin signaling, and endothelin receptor antagonists, which have also been tried in Alport models, seem to stabilize the cytoskeleton in vitro and have some clinical effects. So we may be closer to the clinic than we think with some of these things, even if we're working with very non-specific drugs that we don't quite know the mechanism for. But of course, non-specific drugs could have an advantage because they could work in more patients.
Good point.
Richard Naylor is asking a question. Can I ask you, Liang? So it's a question about the behavior of these synaptopodin knockout podocytes. So are they visibly de-differentiated or more prone to de-differentiation? That is maybe implied by their greater susceptibility to adriamycin, but are there other ways of showing that they're less well differentiated or less stably differentiated?
We didn't see less differentiation.
Yeah.
Yeah.
Not until you hit them with an insult.
Yes.
Yeah, yeah. Oh, that's really interesting. And well, the term in a human disease would be hypomorphic, wouldn't it? A variant, a genetic mutation that doesn't exert any effect on its own, but if another mutation comes along, then it starts behaving worse. Yeah. That's a really interesting angle. And Jeff, I love the way you have people researching on these different things. And you can tell us later whether there are fights in the coffee room or whether there are fights in the coffee room when you're allowed to get near each other. Patrick, should we go on another stop?
Sure. Our final speaker today is Kohei. Would you mind sharing your screen? I hope there aren't fights over coffee in the minor lab. That would be very disappointing. Kohei, we can't quite hear you yet, but we can see the screen. No problem.
Okay. Can you hear me?
Yep, there we go.
I will introduce the basics of nonsense mutation and share what we are doing and part of the result. I'd like to first talk about the basics of nonsense mutation. Our body makes functional protein by reading and translating DNA and RNA as a code. Disrupting the proper protein synthesis machinery causes many diseases. This nonsense mutation is premature termination codon called PTC. It causes stepwise protein production to end earlier it should. This is a common form of mutation in humans that cause shortened and non-functional protein to be made. Let's focus on Alport syndrome. As you know, Alport syndrome is caused by mutation one of any type of collagen, alpha 3, alpha 4, or alpha 5 gene. As you can see here, normal type of collagen alpha chain forms functional complex and becomes a part of glomerular basement membrane. In Alport syndrome, the nonsense mutation induces the shortening of one alpha chain protein. An important point to emphasize that type 4 collagen alpha chain has an essential domain called NC1 on the C terminal. It is an end part of protein to make the complex with other alpha chain. Therefore, the shortened type 4 collagen without NC1 cannot form functional complex and will not be internalized into basement membrane stably. Since shortened type 4 collagen without NC1 is non-functional, inducing full-length protein having NC1 will recover its function and be considered a therapeutic strategy for Alport syndrome caused by nonsense mutation affecting about 6% of patients. To address this, we are focusing on nonsense or PTC read-through therapy. Some small compounds are known to have the activity of PTC read-through, such as aminoglycoside class antibiotics. These compounds are called read-through drugs. The most read-through drugs change protein translational machinery and induce ribosomal skip of premature termination codon. The lead-through drugs enable the nonsense mutant to make full-length protein. For screening nonsense COL4A5 mutants susceptible to lead-through drug, we developed the cell-based screening system in the plastic dish. We use fusion gene, which is a COL4A5 cDNA, fuse inflame to C-terminal non-luciferase gene for monitoring full-length translation. As you can see here, normal alpha-5 chain is translated as full-length form, and C-terminal luciferase protein can make flash of light. On the other hand, nonsense mutant alpha-5 chain is translated as truncated form. This truncated alpha-5 chain doesn't have C-terminal luciferase protein and cannot make light. If lead-through drug induce PTC lead-through, nonsense mutant alpha-5 chain will be changed from truncated form to full-length form. Then they can become capability of making light. So far, we screened 49 nonsense COL4A5 mutation from patients and found 11 mutations were highly responsive to lead-through drugs. This lead-through approach has been getting attention, and some pharmaceutical companies have been studied clinical trial on other genetic disease. So we hope we can translate our basic science research to clinical application in the near future. Thank you for your attention.
Thank you very much, Kohei. A really interesting talk on a potential treatment. Unsurprisingly, I've got a very basic question to ask. What sort of stage would these drugs be introduced in the progression of the disease in order to be effective in preventing its onset? Would we need to introduce drugs to the patient as soon as possible, or could this be for someone that has already lost a fair bit of kidney function?
I think as soon as possible is better, because this approach is targeting to the basics of disease. So if we can induce a stop codon lead-through, the collagen will be normal. So I think as soon as possible is better.
Well, thank you very much. Neil, I'm assuming you have a few questions on that talk as well.
Sure. Kohei, thank you so much. It's really interesting to have gone through all those things which might be relevant to other diseases, and here you are looking at how to alter the mutation itself. There has been a question or two about what the precise effect of these drugs is. So presumably you're, just for if there are any non-biologists out there, just to repeat that this is changing there's a point mistake in these mutations. There's a point mistake in the gene, which instead of changing one amino acid to another amino acid, one building block to another, actually stops the protein entirely. When you treat with these drugs that lead to a read-through, it seems unlikely that they put back the correct amino acid in there. So are you actually creating a mutation, a point mutation, that's just a substituted amino acid in place of a stop codon?
So the changing amino acid depends on the original amino acid.
Yeah.
And sometimes aminoglycoside change from stop codon to arginine. So arginine original arginine stop codon will be the intact collagen. but glycine substituted nonsense mutants will be changed or arginine or some other amino acid. So I think for the first, we are focusing on the non-glycine substituted nonsense mutation.
Yeah, so that was going to be my next question. Can you predict from looking at the mutation whether it's likely to be responsive to a drug?
Yes. By using our system, we can analyze which mutants are response or not. And we found that all mutants are not highly response to leads to drug. Yeah. We so first we screened 49 mutants and found 11 mutants are responsive mutants.
Yeah. And do you know what proportion of Alport mutations are in artificially induced stop codons, these nonsense mutations in humans? It's worth saying these are quite severe mutations. Jeff's waving.
Jeff's got a few numbers. He's trying to hint. Yeah.
I think he's the catcher there. He's trying to indicate a particular ball to throw.
Yeah, I think according to Judy Savage, it's 6%. 6%.
Yeah, yeah. Of which 10% may be responsive with current techniques.
Yeah.
Yeah.
More than 10%. Yeah.
Oh, okay. Yeah, more than at least 10% may be responsive. And the next question is, are any of the compounds that you're using drugs which we already know are safe to use long-term in humans?
In my cell culture study, I use aminoglycoside. It is known that have the kidney toxicity, but now some company developed the non-aminoglycoside compound which has activity of read-through. So we hope they are safer than aminoglycoside.
Yeah, it's always funny to tell nephrologists that one of the possible future treatments for genetic kidney diseases is long-term gentamicin, because all these aminoglycosides are renowned for actually causing kidney damage. In fact, Stephen, they cause tubular damage in particular, don't they? Jeff?
Yeah, I just want to point out that you're absolutely right that some of these drugs might cause toxicity to the kidney, but because of the long half-life of the type 4 collagen, I don't think they would need to be constantly on the drug, because once they make some collagen 4, that should last for maybe weeks to months. And so the treatment could be intermittent.
Jeff,
kind of replenish.
A really important point, and I happen to know that you have done some experiments in the past that show what proportion of protein you need to replace to lessen the severity of Alports.
We weren't that quantitative, but
no.
Yes, we do have some ideas.
Yeah. In other words, you don't need full replacement of normal collagen production rates or permanent production to ameliorate it.
Right. I don't.
Yeah.
Right. I don't think so. No.
Yeah. Yeah. That's really fascinating, Kohei. Thank you very much for that. Let me just
thank you.
Let me just check whether there's anything else in there. Oh, sorry, I'm looking at the answered ones. Yeah, so there's questions again about what the route of administration for these drugs would be. And for aminoglycosides, unfortunately, they are injected, but perhaps some of the compounds you use could be oral.
I think some compound could be oral administration.
Yeah, clearly that would be great. It's always nice if it is a drug that's been used before, but then you're going to ask the question of, is it safe to be on it for years, which for genetic condition is probably what you would want to know. Hence again, why mouse models are so valuable. Rachel Lennon again is asking a really interesting question about when these drugs have been used in other genetic models. What do we know about the long-term issues there, and were any of these matrix disorders?
One is muscular dystrophy, and others are cystic fibrosis. Yeah.
Are any of the cystic fibrosis wonder drugs which are currently on the coming onto the market now, are any of those drugs that help read through stop codons? Or are they just chaperones?
Yeah, currently for cystic fibrosis, like a chemical chaperone class compounds are used.
Yeah, okay. Really exciting. Joy Toll is asking another interesting question about if you can alter it to read through a stop codon, does it have any future applicability towards other missense mutations, this kind of approach?
I don't think so. This stop codon read through compounds can only skip the stop codon. So this approach is not applicable to missense mutation. But previously, I studied the development of screening system for missense mutation to correct hypocollagen folding. So I hope this system will be helpful for developing the therapy for missense mutation.
Reading, just as a general thing, reading through stop codons sounds like a really reckless thing to do. It sounds like it ought to be a bad thing to upset that across the whole body. You know, the stop codons are there for good reason, and if you read through lots of stop codons that aren't that should be there, or that are in there in other molecules, is there concern about other biological effects that this might create theoretically, if it hasn't been studied very much?
I think this stop codon, premature termination codon and normal stop codon is different. A normal stop codon has poly-A tail. So this lead-through drug didn't induce lead-through of normal stop codon.
Doesn't read, yeah.
Yeah.
And maybe there's some protection in, interesting. I'm just watching the time. We said we're going to try and keep to 1.5 hours. Jeff, I'm going to give you a slot to sort of take an overview. And if you, I'm sure you don't, but if you wanted to back a horse in which of these avenues was looking nearest to market, I'm sure there might be some patients interested in which of these routes that you're following in your lab, all of which sound fantastic, which is nearest to actually producing benefit to patients.
Well, that's kind of a loaded question, because of course I don't take any routes that I think will lead to a dead end. So
nearest.
we don't put all our eggs in one basket.
No.
Gosh, that's a great question. So what Kohei didn't mention is that we are maybe he did, but we are collaborating with 2 companies that are providing us their probably phase 1 tested read-through compounds to test in vitro, at least initially, and hopefully move to in vivo. So that has, I think, a lot of promise, at least for certain patients who might have a susceptible nonsense mutation that could be read through. But I think, you know, all these different approaches are important to learn all we can about the different aspects of the podocyte and the basement membrane, and protein folding. And you mentioned chaperones, and I know Konstantinos Deltas is working on chaperone therapy for some of these collagen mutants that don't fold properly. So I think what's critical is that we all research what we think is the way to the treatment or the cure. And it's important that we're all open with each other so that we know what each other, our Alport Syndrome research community, what we're doing. That's why these workshops, I think, are so important. And the in-person meetings, which I hope we'll have next year, are also extremely important, so that our ideas are shared and we get some criticism on why maybe we're not taking the right pathway, to help each other kind of synthesize the best way to finding a treatment and cures and at least hope for patients.
Mm-hmm. Thanks, Jeff. So you're quite near being able to test, at least in mice, perhaps therapy with molecules that might work for a small percentage of human mutations. But as you mentioned, and Kohei, as you pointed out, you can predict responsiveness of mutations to some of these drugs. And in addition to things that read through these artificial or these mutated introduced stop codons, there are other small molecules which might be taken as tablets, which can improve the folding of missense mutations, which another of the questioners raised early. So we really are moving towards a sort of personalized therapy like that, whereas some of the other work we heard about, it sounds as if could be much more broadly relevant to a wide range of patients. So I'd just like to thank you all very much for that really impressive range of work. Back to you, Patrick.
Let's wrap up here. Thanks everyone for joining us today. It's a real inspiration to me as a young patient to see this global community growing and supporting each other, both through research and through you guys engaging with us every week. Thank you to our speakers and to our scientific moderator, Neil Turner, who have put the time in today to talk about their work. This patient-researcher collaboration is, I think, more important than ever during lockdown. And obviously, we can't wait to do physical events once they're allowed. Finally, a massive thank you to Kidney Research UK for your sponsorship. The community really appreciates the contribution you're making to help us understand our condition and connect up such incredible scientists around the world. Before I go, I want to let you know about our next workshop. That's next week on the same day, Wednesday, the 1st of July, at the earlier time of 1 p.m. BST. That is with Kandai Nozu, and they'll be presenting the groundbreaking research on exon skipping therapy, which was made public earlier this month, I think. So absolutely the cutting edge of research on Alport syndrome. It's a real treat to have it presented to us. So make sure you don't miss that talk. Wednesday, the 1st of July at 1 p.m. BST. The week after that, we've got a workshop at 9:30 a.m., which being 22 will mean an early morning for me. And we'll be doing that on eye abnormalities with some presenting friends from Australia. If you have any more feedback about this session or want to be added to our mailing list, email us at research@alport.info. And that should be pasted into chat now by one of our volunteers. You can also follow us on social media. Patients can join the Alport's Warriors Facebook group, and anyone can hit us up on our Twitter page to hear about workshops and other stuff. That's at Alport UK on Twitter, all one word. If you missed anything today or want to have another look at a topic we discussed, we will share the video of our session on the Facebook page and via email sometime in the next week. Wherever you're from, I hope you're coping with lockdown well, and I look forward to seeing you either at our workshops later this month or physically at a proper workshop once the pandemic ends. Thanks everyone for watching, and